And now we'll move into 4 other talks. We'll do 2 and then have another question and answer. So be thinking about those questions, and then 2 more. So first we'll have Chris Somerville, who, based on some of the information you heard about this morning, was really driven to move to Berkeley to become the director of the Energy Biosciences Institute. And he'll tell us about what's driving the research there.
Thanks, Jamie. All right, well, I'd like to just begin with an introduction to the EBI. EBI was founded here about 3 years ago now with a $500 million commitment from BP, and the organization from the founding involves 3 other partners, Lawrence Berkeley Lab, University of Illinois, and UC Berkeley. The mandate of the EBI is not actually entirely biofuels. It's broader than that, and it's to explore the application of modern biological knowledge to the energy sector. And at present, when we began, we already had a pretty clear idea about cellulosic fuels, and that currently is the dominant thing we do. But we have other interests such as petroleum microbiology and biolubricants and biomaterials and pipeline corrosion and other things that we hope to eventually get to. The EBI has, although it was born in quite a bit of controversy here, has actually been a very popular activity when it comes down to participation. My administrators tell me that we've had 770 people on the payroll of the EBI in the past 3 years, including 150 faculty. So if we subtract 150 from the 700, I think that indicates there's about 100 undergraduates and about 400 graduate students and postdocs are or have been involved. So what I'm going to do is give you a little indication of a few slides about what our interests are and some of the things, but of course it's impossible for me to actually describe everything we're doing. It's quite a large activity. One thing I can say, though, is strategically, the way we've approached our mandate in the context, and I'm only going to speak about the cellulosic fuels aspect of it, the way we've approached that mandate is that we could have focused on a few key issues and really put a lot of resources to try and solve, the key issues, but instead, we've taken a broad view of the overall problem, or the overall opportunity, I guess I could call it. So that we are trying to advance a whole system analysis of what might be involved in making next generation biofuels. So we have, as I'll show in a few moments, activities ranging from economics through agronomy and microbiology to chemical engineering. I think I'll go through this somewhat quickly since in view of some of the other talks, but I just did want to point out, remind everybody that energy use is expanding very rapidly, and it's transportation, which is where fuels come in, is actually a fairly small piece of the overall expansion. It's just 20%. So whatever we do in terms of making low carbon fuels is going to be at most a 20% solution. Now, of course, at present, I guess it's widely known here that about 87% of energy use currently comes from fossil fuels. And if we believe the International Energy Agency, in 2030, as you can see, the pie is bigger, but it's approximately the same proportion. So the energy establishment believes that actually the situation is static. And I think based on everything we heard this morning, we should hope that that's not true and that people like the people in this room can fundamentally change this equation. When you ask about how we could change the equation, this shows what DOE at least assessed to be the opportunities to decarbonize energy a few years ago. It's been noted today, that nuclear will decarbonize, but it also was noted earlier today that just to hold emissions where they currently are, we need to build a nuclear power plant every day to soak up the expanded demand because the average nuclear power plant is 1 gigawatt. If we look at through the options on the log scale, this is the 15 terawatts that Chris Field mentioned. And in general terms, the big opportunity is obviously solar. It's 4 orders of magnitude larger than the other opportunities based at least on DOE's assessment. Wind is relatively controversial as to the magnitude. DOE, at least in 2005, was estimating about 4 terawatts worldwide. And of course, that's based on, at least on part, on a calculation of how much persistent wind you need to amortize the cost of installation, and also the proximity to the markets and things like that. This is actually, the motivation. I think long term, it's apparent that as energy use grows, our solutions are going to have to include, to a large extent, solar solutions. So I think of biofuels as a solar solution, as a solar technology. The essential equation shown here is that sunlight is used by the photosynthetic apparatus to reduce carbon dioxide to the components of biomass. And when we burn them directly or indirectly, we can produce work. And the CO2, in theory, cycles round so that you end up with a carbon neutral production of work. In practice, as we know, it's more complicated. For example, tilling the soil stimulates oxidation of carbon stored in the soil by microbes, and that causes off-gassing. Land conversion, meaning if, for example, you cut down a forest and burn everything on the forest unproductively, that, of course, also provides a large negative, a debt, you might call it a carbon debt, that needs to be paid over a long period of time. Fertilizer use, IPCC says about between 1% and 3% of nitrogen fertilizer off-gases as nitrous oxide, which has a greenhouse gas footprint about 250 times CO2. Additionally, 2% of energy worldwide, roughly, is used to make nitrogen fertilizer by that Haber-Bosch process. And then there's all the transportation and processing. So actually, it very much depends on how you do this as to whether it's carbon neutral or even a net gain energetically. So it's and this is certainly one of the reasons why we have a pretty big activity in environmental and life cycle engineering. So today there are most biofuels are made by one of two processes. The most advanced in some respects is the sugarcane process, in which sugar is fermented by the same yeasts used in the beer and wine industry for 5,000 years probably. And then the fuel is distilled to dry it and you produce a product. So Brazil produces today 40% of their transportation fuels on only 4 million hectares of land. The corn process came along a bit more recently, so 65% of a corn grain is starch, and that's just a polymer of glucose. So the process just has one more step, basically, and that is the starch is usually gelatinized by boiling and then converted to sugar, and then the process is the same. Those are mature processes. It doesn't mean there's not opportunities for some innovation there. For example, the drying is currently done by distillation, which is quite energy intensive. And it seems some of the things we work on may ultimately be relevant. For example, it might be possible to develop either liquid-liquid separations for constantly removing fuel from the fermentations, or some membrane process. There's various possibilities to improve some aspects of this. But in general, we don't work on corn and sugarcane. I think we're also pretty skeptical about the long-term opportunity to use food for fuel. So to do cellulosic, which is the next generation fuel, it's actually a highly similar process. In this case, cellulose, like starch, is a polymer of glucose. It just happens to be another isomer. It's an alpha instead of a beta-linked glucan. The overall process is remarkably similar, except, as I'll mention in a few moments, that step turns out to be a little hard, the conversion of the glucan to sugar. Okay, so there's a lot of issues in this, and in the case of cellulosic fuels, probably the most controversial and in some ways the most challenging question is actually not this conversion process, since it's very similar to existing processes, but actually where one is going to get the biomass. And in general, this slide shows something a little bit more complex than that, which is that I think, as I already mentioned, there's lots of complexities in this as to whether it's done well. So in addition, the feedstocks is a big one. That is, depending on how you produce them and how much energy and other goes into the production and the transportation, you can either do it well or you can do it badly. There's a lot of discussion about that. Obviously, agricultural practices trigger an enormous amount of controversy. Let us say the use of nitrogen fertilizer in the corn industry. A lot of that nitrogen fertilizer runs into the Gulf, creates a big dead zone. That's generally considered biofuels done badly. The question is, can it be done better? And so on. There's process optimization, obviously, greenhouse gas and energy balances, economic factors, and ultimately, you have to produce a fuel that meets the goals. So in general, as I mentioned, we're trying to see through all these things simultaneously so that we can determine whether there's really a showstopper in making the next generation fuels. So to do that, we've integrated, on this slide, it's shown 27 different subjects around it. And I don't think I should enumerate them all, but let us say there's a series of questions or disciplines associated with producing feedstocks, the biomaterials. And as you can see, probably that involves everything such as, what we call biotic risk. That is, pests and pathogens can be very destructive. In fact, in Africa and Asia, about 40% of all agricultural product is lost to pests and pathogens. So that's an important efficiency factor that one might not normally associate with biofuels, but actually it might be a make or break as to whether biofuels can really work long run. And of course, there's all kinds of issues with transport and storage and related things. Then once one has feedstock, the next bucket of problems is associated with converting the feedstock to something that can be subsequently converted to a fuel. So there's kind of two baskets of technologies associated with that. And so it leads to actually a very complex series of technologies that have to work together. So to come back to feedstock, I got a bit ahead of myself there. We've established sites around the United States where we're examining different kinds of feedstocks that might be suitable for next generation fuels. This is a part of our farm at the University of Illinois. This is the basketball stadium. This is the University of Illinois at Urbana. We have 320 acres there, just over here. That's a mile by a half a mile, where we've established large plots of candidate species ranging from mostly perennial grasses, but all the way to trees. And in fact, we have relationships with 30 botanical gardens around the world who are sending us candidate species that they think might be interesting. Additionally, we have 19 more sites now around the United States where we're planting candidate species so that we can examine their production, the issues associated with their production, but also their biotic and abiotic stress responses and their effects on the local environment, including soil, water, and gas. Now, there's many species that might be interesting. One of the interesting things about bioenergy is that unlike food, where we've gotten adapted, so all food production in the world is based on 140 species of plants that we've adapted over some tens of thousands of years. But there's 250,000 others that we don't use for food. And one of the nice things about bioenergy is that when you stand back and look at it from first principles, all kinds of new species pop up with good properties. So this is one that's attractive. In the south, this is 1 year's growth of Napier grass. Yields about 45 dry tons per acre year. So you can drive around the world about 4 times on 1 acre of that. And this is a more temperate species. This is a Miscanthus giganteus. This is an 8-year-old plot growing at the University of Illinois. It's cut every year at ground level, and on average we're getting about 18 tons per acre year. with no inputs. So never been fertilized, never been irrigated, or never been sprayed with anything, and shows no decline in productivity over that period of time. So this is very attractive to us for low input, high return. And we've invested quite a lot in this because it has a very broad range all the way from southern Canada to Florida. This is a piece of data about Miscanthus in particular that's unusually interesting to us. What it shows is a 14-year study in England where there was no nitrogen, 60 pounds or 120 pounds per acre of nitrogen applied to the field. And every year the plots where all the biomass was removed. And you can see that over that period of time there was no response to applied nitrogen. And this reflects the fact that these perennial grasses have a growth habit that allows them to withdraw mineral nutrients from the biomass and store it in the ground in a big perennial root during the winter season. So if you simply wait until they've gone through that biological process before you remove the biomass, you don't remove the mineral nutrients, and they recycle each year. So environmentally, it's very attractive. And we believe that very sustainable. There are plots in Europe now that are still yielding after 30 years. To really understand the environmental constraints, we built the biggest and most comprehensive environmental monitoring system in agriculture anywhere in the world. So we have now at several sites, we're measuring everything that can be measured out in the field pretty much. That is all the gas fluxes, including methane. Also soil temperature, moisture, but importantly, we've tiled fields and installed so-called lysimeters where we can measure the mineral nutrient efflux in the water table or in the drainage from these fields. And our goal in doing that is to understand the whole input and output of these systems. And then we've planted all kinds of different potential crops on these fields. So everything from mixed prairie grasses to, we use corn as a control, something we know we don't want to use. So far, the results from these studies, mostly led by an ecologist named Evan DeLucia at Illinois, indicate that the perennial grasses are certainly very attractive in regard to soil carbon accumulation and lack of mineral nutrient runoff compared to conventional crops. So we think that all the evidence to date indicates that these are going to be sustainable and attractive relative to conventional crops. And we actually envision them being used to remediate fields that have been farmed for a long time with conventional agriculture. I'm a little bit rushed for time, but I just want to point out that this is some data from Evan's studies showing that, for example, in this case, switchgrass, where grassland has been converted to a switchgrass, their studies indicate soil accumulation increasing as the years progress. And in fact, we're doing these studies around the United States. One of the issues is whether there's land available, and our analysis suggests, well, I'm going to come to how much land is available. It appears to us that there's relatively large amounts both here and elsewhere in the world. So for example, in the United States, until very recently, we paid farmers not to farm 40 million acres as a mechanism to support the price of crops like corn and soybeans. And this land is actually highly productive, so there's a kind of a myth that somehow this is marginal land, and I just wanted to show that, for example, of the 1 million acres that we pay farmers not to farm in Illinois, the yield on those acres is about 127 bushels per acre versus about 140 for the prime farmland. So there certainly is quite substantial amounts of land available in this country, and I'll come to the world in a moment. And in order to understand how much or which of that land might be suitable for us, an approach we've taken is biophysical modeling of candidate crop species. So we have highly parameterized models that use meteorological data and soil data, that is, insulation, water, wind, and temperature, as well as soil data. And we use 100 biophysical parameters that are able to model the growth of plants now very efficiently. So this is a model for Miscanthus, I think in this case, yeah, Miscanthus, sort of showing where the attractive regions are. And you can see, for example, along the Mississippi and in the southern states, actually turns out to be very attractive. Yeah, one really interesting thing is that you can see in Iowa, there's a place where actually the Miscanthus, well, here it's very high, but here it's very low. So unusual, and we looked into that, and that turned out to be a place where the soil is rather shallow. And it turns out corn, which is the dominant crop there, it thrives on shallow soil, but Miscanthus, as a perennial, has deep roots and doesn't thrive where the soil is shallow. It's kind of interesting. So anyway, this is very useful. This model can be applied everywhere in the world where we're able to get the relevant meteorological and soil data. And it's very helpful for allowing us to see where the opportunities are and to estimate what the likely unfolding of the industry might look like relative to other demands for land and also relative to regional infrastructure and access to markets. In fact, we have a very large team of economists that take the data from the biophysical model and then build logistical and economic models on a spatial basis. So this is based on here's a couple of sort of regional models for several crops, miscanthus, corn, and switchgrass, on a county basis, in this case constructed by Madhu Khanna. And she takes the inputs from our biophysical crop models as a starting point to create her economic and logistical models. And they allow us also to estimate where the good places are for production of biomass and where the bad places are. So you can see, for example, Miscanthus has a much lower cost per ton based on her opportunity cost models than switchgrass. And these are also somewhat important for envisioning what the future might look like. Finally, in the context at least of economics, we have a number of economists and social scientists working with us also to understand the interface between, or the impact of devoting land for the production of biomass rather than food. The long-term trend shown here in food prices is strongly downward, which is really a very important thing to understand, because from there to there, the world population doubled, and yet during that same period of time, all major commodities experienced a price decline. And that's related to, of course, largely to improvements in both the agronomy but also in the genetics of the crop species. So our big question is whether, if we start applying demand for land, whether that's going to impact this long-term trend and what the consequences are for at-risk populations. Our focus at Berkeley, at least, is not America. It's global, so we have similar questions everywhere in the world. We have particularly a focus on Brazil because it represents such a large opportunity. In particular, as I mentioned, at present, the Brazilians get 40% of their transportation fuels on only 4 million hectares of sugarcane. But the Brazilian government recently announced that they would allow the sugarcane to expand to 64 million hectares. So that will take Brazil, if and when they do that, to producing 400% of their transportation fuel. So Brazil, just on that basis alone, will become a very large exporter of fuels. Technology I want to talk about in a moment will allow us to, at present, the Brazilians only use 14% of the mass of the sugarcane to make liquid fuels. They actually burn the rest rather inefficiently where they can and export it to the grid to make electricity where they can. We believe some of the technologies that are coming along, and I think Jay Keasling will talk more about it than I'm going to, will allow us to actually use at least double that. So we believe in the long term Brazil could produce at least 800% of their demand for fuel. Now, it always raises a question as to, well, is that going to impact the forest? And in fact, we have teams of students working down there because we don't think so. It turns out there's 234 million hectares of cattle ranching in Brazil. And in general, the cattle is at very low density, perhaps at least fivefold below the carrying capacity of the land. The Brazilians have used cattle to actually claim land during the land grab. And so we're studying what's required to intensify cattle production to make land available for this projected expansion. And indeed, the government's designation of the 64 million hectares is based on that concept rather than further expansion into the Amazon. These are not things we're participating in. We're just trying to understand it as a factor in understanding the long-term trend. More generally, actually some work from Chris Field's lab a year or so ago showed that based on some satellite imaging they did of land use around the world, that there's about 1 billion acres of agricultural land, land that has been used for agriculture that has been subsequently abandoned. And there's all kinds of reasons for it. In some cases it's desertification or water problems. In other cases, it's in the United States, it's mostly economic reasons for the abandonment. Nevertheless, we're also trying to now get higher resolution understanding of where this land is, why it was abandoned, and what it means. And it's particularly attractive to us because where the land has already been used for agriculture, several things have happened. One is, in general, the carbon costs have been paid. That is, the carbon debt associated with converting an acre of native land to an acre of agriculture was paid sometime in the past. And secondly, in general, that land is not significantly required for biodiversity or ecological activities. Not always true, but it does indicate somewhat the scale of land that may be available. So we'd like to understand that in more detail. Additionally, there's 5 billion acres worldwide that's too arid for agriculture. And so we've become interested in species that will grow on semi-arid land. So this is a very common plant around the Bay Area here, various species of agave. It's usually Agave americana here in the Bay. These species will yield about 10 dry tons per hectare a year on 30 centimeters of rain. And they, as you know, if you just look at the plants in our neighborhood, will survive from approximately April to November without rain, extremely drought hardy. So we actually believe these types of species will be maybe suitable for energy plantations. This is actually a plantation used for making rope. This is Agave cisalana in Africa. So there's about 1 million hectares worldwide of several agave species. So we think that there are options in terms of land and that it's hard to get a real, we don't as yet have not tried to even identify an actual number of to how many gallons of fuel we could make with the land that seems suitable to us by environmental and societal criteria. But right now it seems like there's a very large amount, and so we're progressing. I think I'm running out of time. How much time do I have? A couple of minutes? Okay, I'm really running out of time. Well, we're working on storage and transportation. That's highly relevant. Let me just say, we're also, of course, we have a huge activity in trying to understand how to convert biomass to fuels. And this turns out to be, at one level, straightforward. As I mentioned, the dominant components of biomass, if you look at it up close, are these large fibrils of glucan. Turns out these are very hard to break down enzymatically. The linkages are recalcitrant. So we've been taking all the usual approaches, exploring, looking for new enzymes. And one of the groups in the EBI, Eddie Rubin and a couple of student or postdocs, I think for this project alone, sequenced 280 billion base pairs of DNA. They discovered 27,000 new enzymes for which they have full-length sequence, 27,000 enzymes that are relevant to biomass depolymerization based on similarity to other enzymes. And actually, now our challenge in that program, we think, okay, we have enough diversity now. They increased the world knowledge of cellulitic enzymes, or actually carbohydrate active enzymes, by about 35% themselves. And that was just from one cow rumen. It's kind of interesting, the diversity in one cow rumen, 27,000 novel carbohydrate active enzymes. So now our task is we're trying to work through that and devise intelligent ways to parse that extreme amount of new information into some new understanding of what I would call the the catalysis space, how much interesting diversity is present there. Alex is going to talk about ionic liquids, so I just want to talk about the other remaining challenge. One of the other big challenges we have that we're making some progress on is that biomass is not just made of cellulose, but actually it has a large component of hemicellulose that's made of a sugar generally called xylose, and none of the industrial yeasts currently used will use xylose. So if you look at the cost of making a gallon of cellulosic ethanol where you only use the glucose, NREL calculates it's about $2.40 a gallon. But if you could use most of the xylose, you knock almost $1 a gallon out of it. And that's the scale of effect that we're quite interested in. And so we have colleagues in the organization in EBI that have engineered yeast to use other sugars besides glucose. And some of the results that have come out of this are really are very satisfying because of the way in which we've made the discoveries. So this is from actually one of Jamie Cate's recent papers in Science, and reporting on the discovery of a transporter that takes up, instead of taking up hexose, actually takes up oligosaccharides. And what this is reflecting is biomass is degraded by glycosyl hydrolases to oligosaccharides, and then by a beta-glucosidase to glucose, and that's taken up. And so in the discovery of this class of transporters, he completely removed the need for this pathway. And turns out this is 30% of the cost and 30% of the volume of the enzymes that are used to break down biomass. So even on the basis of that discovery, getting rid of that requirement, this turns out to be a very, very useful result. But the thing I wanted to say about it was the way in which we found it, or the group found it, and it's something like this. We have within the EBI a group that's taking a purely scientific, curiosity-driven approach to the question of how do filamentous fungi, like Neurospora crassa, grow on biomass? After a forest fire, the first thing that grows there is Neurospora, and so the group has gone in, a group of about 11 faculty members and students and postdocs, and simply asked the question, what is every protein made by the organism when it grows on biomass doing? And the transporter popped up from that kind of systematic analysis of how the organism grows on biomass, rather than necessarily trying to solve a specific problem in the overall biofuels chain. And I think what we put in place that enabled this was we have around this kind of basic science group, a group of engineers and people who are knowledgeable about the actual processes involved in making biofuels, who were able to sort of spot interesting discoveries as they've come out of this thing. And so I like it as an example of actually how I think we can bring really great academic basic research to bear on the problem. That is, what we're trying to do in the EBI in summary is support great basic research, but have it aligned with a group of analysts, you might say, who know what might be useful for the full range of problems associated with making cellulosic fuels. And this is for us, an iconic worked example. Well, I think my time is up, if I can understand it, so I think I'll just stop there, and thank you very much.
Thanks, Chris. Okay, now we'll sort of continue with this discussion of how to work with the plant biomass. And Alex Bell, who's been thinking about catalysis for quite a while, will tell us a bit about how we could maybe learn some things from the chemical industry to deal with these very complicated polymers in plants and how those might be converted into useful biofuels.
Thank you, Jamie. Chris has just given us a very nice overview of what biology looks like and what kinds of plants can be used as a source of biomass. I'd like to give you a different perspective, and I'm going to start with an outline for this over here. And what we're going to do is, first of all, give a little bit of a background for why we want to look at biomass as an alternative to petroleum, natural gas, and other fossil sources of energy. We'll take a look at what does biomass look like on the molecular and structural level, and how similar is it to transportation fuels? We'll see that it, in fact, is quite different. We'll look then at several alternatives for converting biomass to fuels, and I'll go into depth into two of the several, and then I'll have some concluding remarks at the end. So I begin by reminding us that the world depends on energy. Every country wants to improve its economic development, the well-being of its society, and it does this in terms of GDP per capita by spending energy. So there's a direct correlation between energy expenditure and the growth in capital or productivity of that group. And you can see that on average, most countries cluster around this yellow line over here. Here's the US at a high level on both axes. But there are several lines here with different slopes. The one here that runs up to Japan represents a more efficient use of energy, and the one over here that runs through some developing countries, developing industrial countries, has a lower energy efficiency. The good news, though, is that if you look over time at the slope for any particular country, it's going from here to there. So we are finding ways, in fact, society is finding ways to maximize the GDP and minimize the amount of energy required to do that. But the world is expanding, and the demand for energy continues to rise. You can see that here from the total. Most of that is coming from the developing nations. This would include China, India, Indonesia, Brazil, and others. The industrialized countries are also increasing their demand for energy, but not at quite the same rate. But overall, there's an increase that today is at about 2%. So over a 10-year period, we're going to increase the demand for energy by 20%. At the same time, the traditional sources of energy for fuels are reaching their peak, as shown here. Here we are at 2010. This is a projection done 6 years ago, so one can question its accuracy. But in fact, if you look at the reports and the projections of when are we going to reach the peak, there is diversity there, but it's sometime between now and maybe 10 to 20 years from now. There's not a big spread in the peak values. So the point is that the traditional sources of carbon and energy stocks that we have used for nearly 100 years will face decline. And it's been estimated by Jeff Sorola, he's a corporate engineer at Tennessee Eastman. It's one of the few companies in the US that uses coal in a large degree to look at producing chemical products like acetates, cellulosic acetates. And he's estimated using the recoverable carbon resources that we have, the traditional ones, how long would they last? And what I want to draw your attention to is the third column here on the right, which looks at the projection based on an expansion in the population and expansion in nations using energy. And so you can see that oil is expected to run out in 25 years, give or take, maybe 5 years, natural gas in 45 years. The quantity in parenthesis accounts for the recent finds of gas in shale, mostly in the US. Coal here is now only good for a century because of the very rapid use of coal, expanding use of coal in China for electrical energy and other purposes. So here's the summary. We've been good using oil, gas, and coal for over 100 years. These have been the traditional sources of carbon for fuels. In the interim, tar sands, shale, and coal, roughly in that order, will be used for the same purposes. They're kind of a bridge. But these first two categories are not sustainable sources of energy or fuel stocks. Once we've used them up and once we've burned them, put the CO2 in the atmosphere, that's it. They're gone forever, as far as we're concerned. It'll take another 1,000,000 years before they're recaptured. Biomass, though, used in a strategic fashion, together with solar energy, has the opportunity for being sustainable. So what I'd like to do in the balance of my time here is guide you through the concept of using biomass to produce transportation fuels. So the basic idea is that we'll let nature take CO2 and water, photons from the sun and convert it into this group over here, which looks like formaldehyde to the chemist, or you can think of it as carbon plus water, i.e. carbohydrate, and it releases oxygen into the atmosphere. So grasses and trees will do this. And what we'd like to do is take this biomass and by one means or another convert it into biofuels, which then are used to fly planes, drive trucks, drive cars. All of it ends up as CO2, but that goes back into the atmosphere and the cycle is continued. Is there enough in the US? Well, Chris Somerville has shown you a map of the US suggesting that there is. Here's a different form of the map from the Department of Energy, which identifies various zones within which certain kinds of biomass grow. And the ones that look particularly attractive are in red boxes, so switchgrass here, hybrid poplars, Miscanthus that has been mentioned, switchgrass again, because these grow with good yields per hectare of land, minimal resources for water, and practically nothing in the way of fertilizer. So today, the US produces about 190 million tons per year of biomass, which is used for energy. Most of it is simply burned. 10% of it is used to make ethanol. But the potential is roughly 6 to 7 times that. So there's a lot. And the DOE estimates that if we were to use this biomass without growing more, there'd be the potential to produce maybe 15 to 30% of the transportation fuels. Remember, in the morning we heard that 70%, 60 to 70% of our transportation fuel, i.e. petroleum, is imported, so we could cut that down perhaps by a third to nearly a half. Biomass, as I'll show you here in the next few minutes, is very different from transportation fuel. So let's take a minute to walk our way through the structure of biomass. So here's a wild grass growing over here. Here are the plant cells. And as you dig deeper and deeper under the microscope and look at what is made up, what's in the walls of these cells, you'll find, first of all, these glucose units. They look like gumdrops over here that are connected together into a polymeric structure. This is called glucan. That's shown over here in the chemical structure. And these then crystallize into these strands. And Chris showed you some of these strands from a micrograph towards the end of his presentation. These are surrounded by hemicellulose, another biopolymer, which I'll show you in a moment. And finally, all of that is wrapped in what I call a wax wrapper, because it doesn't look like a carbohydrate. It's more like a hydrocarbon, and that's the component known as lignin. So here is the structure of the glucan strands, several of them, crystallizing into glucose. It has a hydrogen-to-carbon ratio of 2 and an oxygen-to-carbon ratio of 1. And I'd like you to keep that in mind. Here's the next level of structure. Here's the hemicellulose. It's a different kind of a sugar. It's a 5-carbon sugar, mostly xylose. It's connected chemically by these ester linkages to a fragment of the lignin, but the xylose also looks like a carbohydrate, has about the same composition as glucose. And here's what I call the wax wrapper, lignin. If you look at this structure and you were to look at the structure of lignite, a young coal, they're very closely related. It's an aromatic structure, single aromatic rings with these glycerol-type structures bridging between ether linkages. There's no unique structure here. It's a polymer, highly cross-linked, and what it does is give the plant its stiffness and allow the plant to grow vertically up towards the sun. Now, I'd like you to contrast that with what you want as the end product. Here at the top is gasoline. Here's your pump, 87 to 91 octane. Here's diesel fuel. It's given a rating in terms of a cetane number. So the gold standard for gasoline is isooctane. That, by definition, is 100% octane, and you want something slightly lower than that. But it's a hydrocarbon. There's carbon. These white balls are hydrogen, and it's a branched hydrocarbon. Diesel fuel, on the other hand, wants to be a long, straight-chained alkane. Here's hexadecane, 12 carbons, 26 hydrogens, and it's the gold standard for cetane number. That has a cetane number of 100. So what we're trying to do is take this complex structure and turn it into these simple liquids. These are solids to begin with. These are liquids. It turns out that here, the hydrogen to carbon ratio is the same as here, 2 to 1, but there's practically no oxygen except for the ethanol that's blended into gasoline by law. Diesel fuel has no oxygen. It's a pure hydrocarbon 2 to 1. So clearly, biomass has too much oxygen in it, and in order to get from here to there, we have to take oxygen out. Why so? Well, we would like to have a high energy density in our transportation fuel. You'd like to be able to drive a long distance on a liter of the fuel you put in. So here for reference is gasoline, 35 megajoules per liter. It has no oxygen to speak of. By contrast, at the bottom of the list, if you were to think of glucose as a liquid, I know it is not, but let's think of it that way, it would have an energy density of 14 megajoules per liter. It has 6 oxygens per carbon, so it's at the bottom of the heap. It's partially burned hydrocarbon. You can think of it that way. As you go up the list and you remove oxygen progressively, you can see the rise in the energy value or energy density of the fuel. So one wants to go from here to there, and that means that we have to end up taking out oxygen from the biomass. Now, I mentioned at the beginning that there are lots of different strategies for converting biomass into the final fuels. So one can take the cellulosic biomass, enzymatically break it down into the sugars, the C6 and C5 sugars, ferment them, and produce ethanol and butanol, and there are even options for producing alkanes. And Chris has talked about this, and Jay Keasling will talk some more about it. Another alternative is to add steam and some oxygen and convert all that nice structure that nature made into a mixture of CO and hydrogen, and basically start all over by putting the carbon and the hydrogen together, throwing away the oxygen as water. And that will be done by Fischer-Tropsch synthesis, methanol synthesis, or water gas shift process if you want to make pure hydrogen. That involves chemical catalysis. Another alternative here is to pyrolyze the biomass, which again breaks down all of the structure that nature put into it and produces a bio-oil, which contains chars, alcohols, aldehydes, ketones, aromatics. It's a dog's dish of molecules. And initially, it is quite corrosive and unstable, but if you add hydrogen or take oxygen out, you can create a synthetic oil that can be blended in with petroleum and co-processed. So that's attractive. So what I'm going to do is talk about 2 options. We'll look at this option over here because this is a fairly mature option and see what are the issues. And then we'll look at a variant of going this way, but instead of using enzymatic hydrolysis, we'll do this chemically. We can do it now almost as efficiently as you can enzymatically to release the sugars, and then I'll show you what you can do with the sugars to produce chemicals that can be blended into either gasoline or diesel. So here's what nature does. It produces biomass. One then adds steam at high temperature and takes this basic building block and converts it into a mixture of 3 hydrogens, 1 CO, and you throw away some carbon as CO2. This middle part is known as synthesis gas. But of course, we're completely destroying the chemical structure that nature produced by doing this. The next thing you do is you do this in a large vessel, and I'll show you what a plant looks like in just a moment to produce the syngas. It's quite dirty coming from biomass. You have to clean out the chars. You have to clean out the inorganic materials that come with the biomass. So there's a lot of cleanup that it's done, but it can be done. And you then have a CO and hydrogen mixture that you can combine, make the basic building unit of alkanes, this methylene unit, CH2, throw away the oxygen as water. You can co-produce electric power, so that's good. So you're using your biomass for more than making transportation fuels. The problem is that a lot of char is produced during the gasification, and one needs to control the product distribution of this final step, which is known as Fischer-Tropsch synthesis. To understand that, we can look at what does a Fischer-Tropsch product distribution in fact contain. So here's the molar selectivity of products versus the carbon number of carbon units starting from 1, in this case going up to 20, but in fact the plot extends out to 30 and 40. If we start at the right, here's the diesel range, 9 to 25 carbon atoms, and you want straight-chained alkanes, and that's most of what you get. So this process makes a very good quality, sulfur-free diesel fuel. You look at the gasoline range, which is C4 to C5. You make a certain amount of that, but it's what's called a straight-run gasoline, and it has a low octane value because it's mainly made up of linear alkanes, no branching. So to fix that, what you can do is take this cut over here, which is about 15 to 20% of the total product. These are C2 through C4 olefins and paraffins. Convert the paraffins into olefins and use the olefins. They have double bonds in them. They're unsaturated compounds. And reconstruct them so you can either fold them into the gasoline range or the diesel range. If you're folding them into the gasoline range, what you would like to do is make branched products. And this can be done through oligomerization followed by isomerization. One can also do what is called hydroformylation and hydrogenation and convert these into linear and branched alcohols, such as isobutanol. Isobutanol has an octane number of 109. That's great to blend into something that has an intrinsic octane number of 87. That's how you boost the octane number up to 90. This is mature technology. So here is an example of a plant in South Africa. It's huge. It'll produce 30,000 barrels a day of fuels, gasoline and diesel. It costs $1 million at this scale. Now, to put this into perspective, the Chevron refinery in Richmond, just up the road from us here, is a 250,000 barrel a day plant. So think of this as an order of magnitude. So if you want to replace the Chevron refinery with something similar to what the South Africans have done, this is a $10 billion investment. And you don't have to do it once, you have to do it multiple times. So this is expensive technology, and it's made more expensive by the fact that the biomass gasification is more expensive per unit of synthesis gas than it would be if you were using natural gas as the starting material. Okay, I want to turn next to an alternative, which is going this way, but purely chemically. So what we want to do is find a magic solvent that will take all components of biomass and put it into a fluid phase. So we want to take a solid and dissolve it. It's not unlike taking sugar, putting it into water, and it dissolves, and then you can process it. So here's a cartoon of biomass. We want to dissolve it, and I'll show you how we can do that. We then want to break up the carbohydrate into the individual sugars, for example, glucose. We want to take the oxygen out of glucose, remove the oxygen, but keep the carbon and hydrogen in, and ultimately make a fuel. So the challenge here is to find a solvent that will dissolve everything, a way to hydrolyze, a way to deoxygenate, and all of this will involve finding a proper solvent and compatible catalysts here. Well, it turns out that in the last 6 years or so, it's been discovered that ionic liquids, a rather exotic type of solvent, will dissolve virtually every component present in biomass. So what's an ionic liquid? It's a salt made of an organic cation, the positive charge, which is element, which is shown here, and an inorganic anion, for example, the chloride anion. And these are materials that become liquid at 100 degrees C or below. And that's why they're called ionic liquids. They're purely ionic. There's no water in them or anything else. But they're powerful solvents, and they will dissolve cellulose, hemicellulose, and lignin, the principal components of biomass. Why do they work? Well, we've been studying this, doing, together with Baron Smith and Jiwei Chu, statistical mechanical investigations of how ionic liquids interact with components in biomass. And here I show you crystalline cellulose. It's a compact structure. It won't dissolve in water, but it does dissolve in an ionic liquid, and it does so because the small chloride anions can get in and interfere with the hydrogen bonding here, shown in red and green circles, that keeps this crystal together. So we disrupt the forces that keep the crystal together. The larger cations, in this case, ethyl methyl imidazolium cation, squeezes in where the wedge has been placed by the chloride and expands the structure, and you end up with a nice, clear, yellow-colored liquid. So once we've dissolved the crystalline cellulose and gotten these glucan strands, the next thing to do is cut the ether linkages and release the glucose. And this can be done by adding water, just enough water to compensate for the breaking of the bridges over here. So we release the glucose over here. And that can be done with a variety of acids, even very garden variety acids like hydrochloric and sulfuric acid. And here I show you an example of how far you can go. This is done taking crystalline cellulose, commercial name is Avicil, the catalyst to hydrogen chloride, the solvent is emim chloride, an ionic liquid. And you can get roughly 70% of the starting cellulose back as the sugar dissolved in the ionic liquid. There's a little bit of the dimer of that sugar, known as cellobiose, and there's just a trace, about 10%, of what is called HMF, hydroxymethylfurfural, which is the first dehydration product. I'll show that to you in a moment. So you can recover much of the sugar that is present in the cellulose by dissolving it and hydrolyzing it. You can do the same thing to the same extent using hemicellulose. In fact, recently we've discovered that we can take miscanthus in the form of very small particles that are ground up, throw it into the same ionic liquid, add the acid, and we can get glucose, and xylose and arabinose, the sugars that you get from hemicellulose and cellulose, a little bit of the dehydration products of each of these sugars. The net effect is that until recently we could get 53% of the cellulose and hemicellulose that starts out in the material, that's right, that material right there, into solution. And today, by other tricks that we've learned to play, we can get this up to 70, and we're marching up towards 80%. So we can get a large part of the carbohydrate into solution. Well, once you've done that and you have your sugar in the ionic liquid, what do you want to do with it? Well, obviously, there's too much oxygen here. You want to take that out. The easy thing is to dehydrate it, and that will happen spontaneously. The thermodynamics are very much in favor of taking out 3 of the 6 oxygens. So you get this product, 5-hydroxymethylfurfural, or known more familiarly as just HMF, with about 95 or 95 or better percent selectivity. So that's step 1. But unfortunately, this is not a fuel candidate. It has chemical properties that are not compatible with putting it into gasoline. But you can start with that product, add hydrogen at elevated temperature in the ionic liquid, and take this product into a variety of products here, the most desirable of which is this dimethylfuran. Notice now I started out with 3 oxygens. I have 1 oxygen here. I started out with a ring over here. I've kept all the carbons in place. I didn't lose any carbon. So why is this attractive? Well, if I look at my scale of energy densities, there's 2,5-dimethylfuran. It's just a tad below gasoline in terms of its energy density. It also happens to have a low vapor pressure, so it won't evaporate. It meets the specifications that way. It doesn't have any functional groups that will interact with the seals in your engine and cause them to leak, so that's acceptable. What's not clear is how safe is it to burn. It's not your garden variety fuel. Therefore, the energy companies don't know exactly whether it's toxic or not, or whether the products of combustion are toxic or not, and this is a subject that is being investigated. But there's a lot of interest in this material. Notice that there were other byproducts. Are any of them good for anything? Or should we just throw them away or burn them? Or can we produce something interesting from them? Well, it turns out that we can take these products and combine them in strategic fashions by one of 2 chemical reactions. One is called etherification. So here we have this methylfurfural alcohol, which is one of the byproducts. We can take ethanol, which you can certainly produce from biomass, combine it to make this 2-ethoxymethyl 5-methyl furan. It's an ether. Here's the ether linkage. And it turns out that there are patents already today for this molecule's use as a diesel fuel. It has a good cetane number. It has good burning characteristics. One can also make hexane dione, which you can use, and this dimer, which might also be a candidate diesel fuel. The hexane dione, which you produce here, and also as a byproduct of hydrogenating the HMF, can be coupled with methyl furfural by something called aldol condensation to make these longer chain bridges. If you add hydrogen, you can knock out the oxygens over here, which you don't desire as ketones because they swell the engine seals. And now you've made something that has multiple carbon atoms and, again, is within the diesel range. So our sponsors like these molecules. So what I've shown you is there's a wide diversity here of approaches. I've only talked about two. One which is mature but expensive, another one which I will call immature, unclear whether it will the baby will make it all the way to the marketplace and be used, but if you don't grow babies, you never discover whether they're going to be geniuses and get Nobel Prizes. So we're growing babies and seeing what they mature into. So I'd like to end by showing that I've talked about this end of the cycle. In fact, this is the picture same picture that Chris used at the beginning of his talk about the opportunities for converting biomass into fuels. But there are other challenges. One is, how do you grow disease-resistant crops in a sustainable manner so that you don't disturb the soil, you don't release extra CO2, you use the minimum amount of water, preferably no fertilizer. And that can be done. There's also what I call the collection issue and transportation issue. Farms are distributed over many hectares. We need to collect the raw miscanthus, shown here as bales, and transport it to the place where the production will be done. Possibly we'll have distributed production. It won't be done in one gigantic plant the way we process petroleum. There may be many smaller plants, much in the way that milk is processed from milk farms into cheese eventually by collecting and collecting again at various points. But the bottom line is that in principle, it should be possible to go around the cycle in a sustainable fashion. So this brings me to the end of my talk. I'd like to, of course, acknowledge the Energy Biosciences Institute that is providing the support for my own research and much of what I've talked about, and of course the funding from the original source, BP. Thank you very much for your attention.
Okay, so we'll take just a few quick questions, and then we'll have a short break. So we'll start with just a few questions.
And I think the questions are for both Chris and myself,
For both
right?
Chris Somerville and for Alex.
Yeah. So Chris, do you want to join me up here?
The chemical process that you just described was for Miscanthus. Could it be used for other things like sweet sorghum or biofuel or sugarcane or switchgrass?
Yes. I mean, to the extent that you're working with a carbohydrate, the exact source of the sugars ultimately doesn't matter. It might be easier or harder to dissolve or process the starting material depending on what it is, but in principle you can do the same chemistry.
Ability of the salt solution, do you regain it easily, or?
Yes, this is the $64,000 question, which I'm asked by Chris and Paul Willems all the time. We're talking right now with several manufacturers, commercial manufacturers of ionic liquids. They believe that they can develop the technologies to do this. But it's an open question, and it's absolutely essential that it be resolved because these are materials that today are specialty chemicals, but like many things in their infancy, they're expensive, but if they were, in fact, there were demand, commercial demand, their price would come down. There are some ionic liquids, in fact, you can make starting from glucose. So you might use the biomass as your source of the cation. But it really remains to be seen how well you can recycle. And you have to recycle much better than 99.9% of what you put in. So there is even a concept one manufacturer suggested, rent your ionic liquid, like you rent platinum catalysts in a refinery. You ship it back, hopefully not too often. They reprocess it and give you a fresh batch.
Okay, let's take one more question.
Have you done any experiments related to Miscanthus in China? Because I often heard that in China, we heard some lack of electrical coal.
You want to answer that?
We've not done anything in China, but as you probably know, Miscanthus is native to China. That's the source of origin. So all of the clones, all the material we have originated in China. I believe it's used for making paper in China, actually. But we have not done anything there.
Okay, well, why don't we thank the speakers? We're going to take a 5-minute break.